All right. Good evening, everyone, and welcome to the Med School Mentor Leaders in Medicine webinar series. My name is Chin Ken. I'm the founder of Med School Mentor, and from our Med School Mentor team, we have Mia, Cami, and Pedro joining us today. And today we are honored to be joined with Dr. Michael Platten, who is the professor and chairman of Neurology at the University Hospital Mannheim, head of Neuroimmunology and brain tumor immunology at the German Cancer Research Center, and the president of the European Association of Neuro-Oncology. Throughout his career, Dr. Platten has made groundbreaking contributions to how glioma interact with and evade the immune system. His research has led to the development of novel therapeutic vaccines targeting tumor-specific mutations and opening new avenues for personalized treatments in malignant brain tumors. Beyond his scientific achievements, he is an influential leader in global neuro-oncology, shaping both research and clinical practice across Europe and beyond. Dr. Platten, thank you so much for joining us today. I believe you have a brief presentation for us. The stage is yours.
Okay, thank you very much. So first of all, thanks very much for the invitation, and I'm happy to share a couple of thoughts and in a brief presentation illustrating my journey as a clinician scientist and discuss with you how this impacted my career and what the learning lessons are. So let me see if this works. So as already introduced, I'm a clinician scientist. I have two homes. One is the University Hospital Mannheim, which belongs to the Heidelberg University, and then the German Cancer Research Center. So lab work and clinical work, more or less. But I would like to show you that there is a team of mostly basic researchers, but also clinician scientists working in the lab. There's a clinical team working in the clinic, but there's a large intersection between those teams. And I think that this is important for moving forward. So let's go back how I started when I was a medical student. And I understand that the systems in Germany and in Europe and the US are a bit different, but this is how I got exposed in my pre-radiation era as a medical student to science when I got interested in neuropathology and joined the team of Andreas von Deimling, a world-renowned neuropathologist, and worked at that time with very simple methods to identify polymorphisms in a specific types of gliomas and analyzed a large cohort. And this was, for me, a really important experience, not only because I got introduced to science, not only because I published my first paper, but even more to get access to a way of scientific thinking and also a network, which then brought me to David Lewis, who is now chair of pathology at the MGH. And I spent a rotation in Boston working on a different project, again, staying with brain tumors, brain tumor genetics, worked on pilocytic astrocytomas and identified an alteration in the gene for neurofibromatosis type 1. And got to publish my second paper. And this was an entirely different experience. I also had the chance to do a clinical rotation in neuro-oncology. And based on these experiences, the decision was firm to stay in that area and continue my career as a clinician scientist. I then started my residency at the University Hospital of Tübingen, at that time joining Michael Weller, and my work stayed within the realm of neuro-oncology and expanded more into fundamental immunobiological questions revolving around how the immune system deals with brain tumors, working with animal models as shown here, and really broadened my horizon with respect to how tumors interact with the immune system. And the key question, I think, at that time was, do I perceive myself as a basic scientist, as a clinician, or as a clinician scientist? But what's more important is the question, where is the clinician scientist position? And at that time, my perception was more or less, it's in between two worlds, but it's probably not at par, neither with the basic scientist or the clinician. But in order to combine both worlds, you have to lower your standards. And throughout my career and the further steps, I will illustrate that this is not true. You can have a bigger ambition than that. So the question, during my residency, and then the desire to follow a scientific track, was then where to do your postdoc. And there are different types of questions which you can ask is, do I want to follow a certain type of technology, enter a new field? There's certainly the celebrity factor with respect to a renowned lab that you like to choose, different type of culture, and several questions that you can ask yourself with respect to what do you expect from your postdoc. My specific expectation was, if I want to understand how the immune system deals with a tumor like glioblastoma, I need to understand how the immune system works in the brain and, for instance, causes neuroinflammation. So the decision was, I need to understand better diseases such as multiple sclerosis in order to understand how the immune system can potentially be used in order to fight tumors. That was a simple view, but there are a lot of analogies. So the decision I made at that time was to join the lab of Larry Steinman at Stanford University, a fantastic neuroimmunologist at that time, and work with him on a very specific project that revolves around what are immunomodulatory mechanisms that protect us from multiple sclerosis. And at that time, we identified a gene that is intricately involved in regulating immune responses, and also identify a drug that specifically modulates this indolamine 2,3 dioxygenase. Made some animal experiments and published a nice paper from that time. But what is more important, and what I learned, and that was a big learning lesson for me, was you can be a clinician scientist, and at the same time you can be at par with basic science and also with clinicians. So you don't necessarily have to lower your standards. And I think that that's a very important learning lesson for me as someone who was interested to become ultimately a clinician, but not give up on science. Oftentimes then, after such a postdoc experience, the question is, what do you do? Do you leave academia? Do you transition to industry? Do you want to become a junior research group leader, finish your residency? So this is all, again, sort of a crossroad. What I decided at that time is to go back to finish my residency, and I was fortunate enough to meet at that time two wonderful medical students, Christiane Opitz, who has already finished her medical school at that time and was a young resident, and Tobias Lanz, whom I got interested in working with me on several projects that were following my postdoc work. And again, this was a very fortunate encounter meeting these two. And if I look back now on how they have developed and their careers have developed, they essentially chose a different path in that, although both completing medical school and at least starting their residency, they have chosen an entirely scientific career. Tobias is now professor of immunology at Stanford University, working on multiple sclerosis. Christiane is a professor of neurology and leading her own research group at the German Cancer Research Center. And they both have evolved and are now very successful driving their own fields. So these are alternative career paths, which are not clinician scientists, but really evolving to scientists, and that's perfectly okay. So for me, the question was then, do I continue after the residency as a senior physician? Do I focus on clinical work, subspecialization? Again, there's always the question lingering around about leaving academia. And I was fortunate to then transition with Wolfgang Wick to Heidelberg at that time to found a new department of Neuro-Oncology, had my own junior research group. Christiane and Tobias at that time joined me. My first PhD student, Ulrike Litzenburger, then joined me as well. And we were continuing our work on tryptophan metabolism and its function in glioblastoma. And there, again, another learning lesson for me was to be able to be patient in driving your own first work as an independent junior research group leader. At that time, we defined a new tryptophan metabolizing enzyme, TDO, to be involved in regulating immune responses in glioblastoma. Accrued a lot of very interesting data, which would have made a nice publication, but we decided to take this forward, ask more questions, and really then dissecting the mechanism of how the effector function of this enzyme is and how this pathway altogether is regulated with broader implications than just focusing on glioblastoma. And this was a very good learning lesson for me, to take your time starting your own research group until you finish a paper, and take your time in asking specific questions. And then from 2009 on, there was sort of a readjusting of the focus, entirely focusing on glioblastoma research. Was fortunate to meet Teresa Bunse, a PhD, and Lukas Bunse, now a very successful clinician scientist, still in my department. And we were at that time asking the question, can we use antigens that are specifically expressed in glioma and that serve as driver mutations, as sort of neoantigens in order to develop immunotherapies. At this time, the mutation in IDH1 was just discovered. Andreas von Deining has generated an antibody. So we knew that this is a mutation that is in principle amendable to a specific immune response. And again, here we started to test and did different types of assays to ask the question, is it seen by the immune system? And sort of this was this first set of negative data, and this project was almost killed because this was an antigen that was obviously not presented as we believed on MHC class 1, and luckily we continued our work and saw that this is an antigen that is presented on MHC class 2 molecules, and continued our work. And these are essentially the two decisive experiments that Teresa and Lukas did to show that the immune system in a patient is capable of differentiating between the mutated and the wild-type antigen, and a decisive animal experiment where we generated a long peptide vaccine that is capable of controlling tumors in syngeneic mice. So these were the decisive experiments. And then the question is, as a clinician scientist, what do you do with these experiments, except for projecting that this can be potentially used as a therapy in patients? And this is sort of the trajectory summarized of what we did with the first start of the project. Some initial funding, the discovery, the patent application, is really moving this forward to a trial concept and the initiation of a trial within a very short period of time. And this is probably the most gratifying experience that you can have as a clinician scientist. So we ran a multi-center phase 1 clinical trial where we tested for the first time this long peptide vaccine in a cohort of a little over 30 patients, where the vaccine was integrated into standard of care. And again, at that time, the way the trial was set up, that we were accompanying this trial with a very thorough biomarker program And the data that we derived from this biomarker program with that trial, we still exploit as of today, although the trial has finished and was published in 2021. So again, a very scientific attitude when planning this clinical trial and conducting this clinical trial. So this is the outcome of the clinical trial, and we have published 3-year progression-free survival and overall survival. And then the question is, okay, you have accomplished your translation. We have just accrued funding for follow-up phase 2 clinical trial that then will be randomized. But again, for us, for me, the question is not only can we deliver this to patients. This is a very important, maybe the most important question. But what are scientific questions that are still unanswered? And these are, at least with respect to this type of vaccine, and we don't have to go into detail on everyone. All of these questions is, what are the research questions that are still unanswered when we think about vaccines for brain tumors or for cancer in general? And you can see that there are a lot of questions that are still unresolved, and we can use the brain power, new methodologies, and probably we have to develop new methodologies in order to answer these fundamental questions. At the end of the day, the answers that we will derive from these questions will not only help us further understand what we're doing, but then ultimately develop new therapies. So the overarching thinking behind all of this, and this is something that we do to date and will be probably doing in the future, is perceive the translation of new discoveries into clinical application, not as a linear process, but rather as an iterative cycle of translation, where we start with an early clinical trial, as indicated by the IDH vaccine. We try to collect as many biomarkers as possible and do research with these biomarkers, and have very specific questions that we would like to address. And these answers will then help us to optimize the treatment and then feed that back into the next clinical trial. And this is what we're doing. These are just some examples of the clinical trials that we have initiated, shown here, but also methodologies that we have developed along the way in order to better understand, in this case, the breadth of immune reactivity to brain tumors. Of course, this is something that can only work if you're immersed in a research and clinical network. And I'm fortunate to be immersed in such a network here in Heidelberg and Mannheim, which is the National Center for Tumor Diseases. Also the German Cancer Consortium with several sites whom we collaborate with and whom we run the clinical trial with, and a very strong clinical trials office, but also programs like a very active clinician scientist program that helps us to recruit new talents that help us address these research questions and run the clinical trials that we're running. And with that, I'm at the end of just sketching out my personal journey, the decision points and the crossroads that I was in, the decisions that I made that allowed me to be where I am today, which would not be possible without a fantastic team of basic researchers and scientists. And maybe as a basis and to spark of the discussions, my personal 10 learning lessons from this journey. The first one I actually stole from Mark Davis, which at Stanford University, which is essentially don't follow the crowd. I think it's important to always be curious, to enable creativity, to think outside the box, which also then comes with the second learning lesson, to be bold. But if you're bold, always keep in mind that you need to mitigate your research risks. I think that when you do research, it ultimately always comes with mistakes, also when you're a clinician. You need to admit these mistakes and then seek alternatives. What I have learned over the years, also when mentoring a lot of people, is that oftentimes writing down your achievements, writing a paper, writing a grant proposal seems to be a big roadblock. So this is something that early on is a big learning lesson. And for me, one of the biggest gratifications was when I wrote my first paper as a medical student when I was in Boston working with David Lewis. And he essentially said to me, why don't you write a paper draft? At first I was overwhelmed, but then I started doing this. And the experience that you can become an expert at that early stage of your career, if you just lead the literature, ask scientific questions, conduct experiments, is a very gratifying experience. Mentors, and I've showed some of my mentors, are very important. I think that one big learning lesson is that opportunities will come. It's just a matter of seeding them. So my career was not pre-planned, and oftentimes it was just opportunities that I just seized rather than making very specific long-term decisions. It's important to embrace and drive innovation and to keep this mindset. And when you stay in academic medicine and are interested in translation, then don't stop just with translation. The process of reverse translation, i.e. taking scientific questions from the bedside back to the bench, is very important to me. And I think that this is also something that is very important when you strive to develop new therapeutic treatments. Always try to ask questions. When you're asking questions, expect to be wrong and admit that you can be wrong. This is part of the scientific process. And one thing that I learned early on, and this is why I showed the examples of Christiane Opitz and Tobias Lanz, it's very important to become a mentor, even at early stages of your career. But when you're doing so, try to empower your mentees and not sit on the results, but give them visibility. That for me was a very important learning lesson that was shaping my career. And I try my best in order to be such a mentor that also empowers mentees. And with that, I'm at the end. I stop sharing my screen and I'm happy to enter into the discussion.
Thank you very much, Dr. Platten. That was very nice. Thank you for walking us through your career. I think we always like to start our questions here with asking you a little bit about, you mentioned how during med school you're already involved in research on pathology and neuropathology more specifically. I guess my question is, before going into residency and kickstarting your research career, what was Michael Platten before the Dr. Platten era? And did you always know that you wanted to go towards the neuroscience side of things, or how did that decision go in your mind?
So I was always interested in neurosciences from a perspective that I liked the idea that the nervous system is complex, but also very much in order. And from a clinical perspective, you can identify lesions in the nervous system by just looking at a patient and do a neuro exam. This was always fascinating to me. Why I stumbled into neuropathology, honestly, I was sitting in a pathology lecture, and for whatever reason, it was just 4 of us. And then Andreas von Deimling came into the lecture room and was looking around and saying, I'm not giving a lecture for 4 students. You can either go home or come with me to the lab. And I found this so fascinating that I joined him and went to the lab. And this is, what I indicated. It's seizing an opportunity. I could have gone home, but I found this very interesting, and I joined him, and I was exposed to his research lab, which was fascinating to me. So this was honestly the entree into neuro-oncology, to be honest. I wouldn't want to become a neuropathologist because I liked working with patients. So the synthesis of becoming interested in research questions revolving around brain tumors and wanting to work with patients was then clinical neuro-oncology.
Perfect. Thank you. So Mia is gonna ask you the next question. She's having trouble with unmuting, I think, the microphone. So I think Camilla, can you help? Can you help unmute? Yeah. Sorry, sir. We are having some technical difficulties.
It doesn't let me to unmute her, only to ask her to mute.
See. All right. So I guess while we troubleshoot this, can we carry on with the questions?
Yes. So, Dr. Platten, mentorship can make a big difference early in someone's career, but finding the right mentor and knowing how to approach them isn't always easy for students or trainees. From your perspective, what is the best way for someone at that stage to identify potential mentors, approach them in a meaningful way, and build a strong and lasting relationship that truly supports their growth?
It's a complicated question and it's a big ask. So maybe I start with what is not going to be successful. I'm very often approached by students with applications, to join for an internship, a practical, and so on and so forth. And when I get the impression that the requests are generic, that I'm just one of very many addressees of this email, if I don't get the impression that there's a very specific reason why I am approached, and also if I get the impression that it's written with the intention just to gain something and not to bring something to the table, I'm less inclined in responding or accepting that student. So whenever you approach someone who you think could potentially be a mentor, it's always important to make it personalized and to give specific reasons what you want to do, and also offer what you bring to the table, because this is always a mutual relationship. How do you identify a good mentor? My impression is always when I get approached by students here in this ecosystem is they've heard from other students, from people working with me that this is an interesting environment to work with, and a good environment. And whenever I'm interviewing a student, what I always do is introduce him or her to the lab and say, ask the people in the lab, is this a suitable place to get a good impression? So before embarking on such a position, I would always advise anyone to, take a day, maybe 2 days, sometimes a week, to get to know the people, for instance, in the lab or in the department, and talk to them and get a good impression of what this environment and their specific mentor is like. If it's a good mentor, he or she will create a good working environment where people like to be, spend time, and work on interesting projects. And then sometimes you need to be persistent and seek unconventional routes of communication and contact. And in an area where we are so overwhelmed with emails, this is probably not a good way to approach someone. Sometimes pick up the phone or even show up and see if you can get someone. What works best in my opinion is if you go to, for instance, a lecture or a scientific presentation at a conference. And then after that presentation, if there's someone you think would be interesting, just go approach saying, fantastic presentation, I have a couple of questions. And then you can enter into a discussion and saying, would there be a possibility of me joining your department lab for a little while? So these are, more general advices of how to choose your mentor. But the basic principle is really, what would you like to achieve and what would you like to do? And this is the first and most important question.
Thank you so much for the advice.
Yes, that was great, Dr. Platten. I cannot overstate how great it is to hear these things. We are a good part of our community is med students and pre-med students who are trying to figure out how to enter the field, and that it's extremely helpful, that response and going to that to those lengths. We appreciate that very much. I guess the next question is, you mentioned in your presentation how you were worried about how your pursuit of being a clinician scientist and whether it might hinder your development if you were just a basic scientist or just a clinician, and trying to merge those two scenarios. And I guess my question then is, as a clinician scientist, how has research changed the way that you practice medicine in your clinical practice?
Very much so. I think that if you want to thrive as a clinician scientist and you want to have the ambition to make fundamental discoveries and be a good scientist, which is not easy, then you have to accept that you specialize in your clinical practice, right? For instance, I'm chairman of the Department of Neurology. I'm also overseeing, stroke service, but I do as an outpatient clinic mostly neuro-oncology patients. This field is a good example where many patients seek experimental treatments just because there is only a limited number of approved treatments for many of the brain tumor patients. So very quickly in the consultation, you get to a point where you discuss experimental treatments. You explain experimental treatments, how they work. Many patients ask very specific questions. And then you almost enter scientific discussions with the patients. So, being a clinician scientist immediately then translates into how you interact with patients. And patients realize this, and they also appreciate these types of discussions that you have with them. And then, of course, the clinical trials that we're conducting, right? What we can offer to patients, new experimental treatments, multi-center clinical trials that we're running. So this obviously has a big impact on our daily, or my daily clinical work. I wouldn't want to miss this if I was if I was asked, you need to drop either or, it would be a very difficult decision. I wouldn't want to miss either of this, but I'm certain that not only has the science that I've done, that we do, impacted and still impact the way I practice medicine, but also the other way around. I think that one of the best experiences that you can have as a clinician is working with patients, is deriving research questions from clinical problems. And I think that this is one of the advantages that we as clinician scientists have, is that we can derive research questions from our clinical practice. So not only biomarker samples and tissue and blood, but really research questions from clinical problems. then translate them back to the bench. I think that this is a very important part of daily practice, at least for us. You need to work with outstanding, smart, basic scientists. But in my interactions with many of the PhD students, I sense in many of the PhD students who want to do excellent basic science, also an urge to put meaning behind the basic science that they're doing. So they very much appreciate that what they're doing, the research that they're doing, the questions that they're addressing, that they ultimately at some point may become available for a treatment for patients. So this sense of giving a broader meaning with respect to helping patients to the science is a motivation for many of the basic scientists that I work with.
Makes sense. Thank you.
Dr. Platten, thank you so much for your time and your patience with the reboot. I was wondering, translational research can be incredibly exciting, but also slow and challenging. How do you personally balance ambitious, high-impact ideas with also practical realities of funding, regulatory hurdles, and timeline and patient outcomes?
That's a very difficult question. Again, going back, I think that if you develop an idea and want to pursue this idea, there are many hurdles. These are regulatory hurdles. Oftentimes it's slow, as you indicated. But what has helped me and us a lot is try to assemble a team and have the many tasks that are necessary in order to accrue funding, to write a clinical trial protocol, to discuss with the regulatory authorities, see this as a team approach. And this is something that I try to do from the very beginning. It goes back to what I mentioned, is empower your mentees, is trying to put people in the position to help, not only help with the process, but maybe also own parts of the process. The task, for instance, conceptualizing and then running a clinical trial is so big that this is not something that a single person can do, but it's always a team effort. So this is probably the biggest advice. You need to accept that there are pushbacks and things take longer. This then goes back to this high risk types of projects. So you need to have a risk mitigation strategy, maybe set intermediate goals that you can fulfill, accrue funding for sort of side projects. For instance, what we do when we get a clinical trial approved is have specific research questions that are attached to the clinical trial and seek independent funding for these research questions. So these are some of the considerations. And then last not least, I think that the research environment is very important. Do you work at a place where this type of approach, this translational approach, is supported, or where there are difficulties inherent to this place? At least in Germany, there are only very few places where this could be done. You can be as smart as you want to be and as energetic and as creative. If you're not at the right place to be able to do this, then you will probably fail. It's similar to in the US. I know this, and one piece of advice that I always give is, in your career, if you advance and you get offered positions, oftentimes there's a tendency to accept a position at a certain institution where you may be the only one and the leading figure in a certain field, for instance, in neuro-oncology. If you want to translate something, it's good to be in an environment where you have many peers, sort of in general focusing on the same goal, for instance, neuro-oncology, and not perceive them as competition, but rather as a stimulating environment where you can benefit from and that helps you achieve your goal.
Lovely. Thank you for your input. And I'll be passing it on to Cami for our next question.
Thank you. We have a question from the audience. Umar, you can open your mic and ask the question directly. Okay, I will read it for him. Could you share some thoughts on how an international student should approach a professor, scientist for a PhD in neuro-oncology field? Also, could you give some advice for a student who wants to join a PhD? How can they prepare themselves?
So in essence, when I receive applications for PhD positions, I usually direct them to a central program of a graduate school that we have, which has the advantage that applications are pre-selected and then candidates are invited for an interview. This is different from some of the other places where individual applications to professors and scientists are then evaluated and invitations are issued for an interview. As said, coming back to approaching a mentor, the biggest task is how do you stand out from the crowd when you submit an application. And again, non-generic, why specifically you're interested in that group or that specific field, what you bring to the table, what you would like to achieve. So these are very important aspects of such an application. Oftentimes you will not hear a response, then it's good to follow up. And as I said, seek unconventional ways of approaching a professor or scientist you really are interested in, right? For instance, see if you can, seek someone at an international conference, and this is, oftentimes a good way to approach. If you want to prepare yourself, again, I think that the mindset is probably more important. Of course, for certain PhD positions, it's good to be able to handle certain types of methodologies. In these times, I advise every medical student and every PhD to have a good understanding of bioinformatics, know at least to handle and exploit large language models, because this will be the future. In all your scientific career, you will experience that the individual experiment as such will be of less and less importance. But your scientific question will be more important, and your question that you can potentially address with existing data will be more important. So this is probably a way to prepare yourself. This is still a bottleneck, analyzing large sets of data. So it's a mixture of advices that I can give, and then it very much depends on what you would exactly like to achieve in the field of neuro-oncology. For instance, if I receive applications from someone who is interested in the role of glioma stem cells, period, I would probably send this back and answer, I'm not the right place, I'm not the right person, I'm not glioma stem cell expert, there are different labs who you should seek. So it's good to be very specific in what you would like to achieve in your PhD.
Thank you. And also another question, Dr. Platten, over the years you have taken on major leadership roles, including your current position as president of the EANO. Earlier in your presentation, you highlight how much institutions and collaboration truly matter, how progress often depends on the strength of the teams. Could you elaborate more on this? And could you also expand on other qualities that truly define a strong and effective leader in academic medicine?
So first of all, I think that I never had the goal to be a strong and effective leader in medicine. This sort of comes more with growing into a field and having the ambition to move science forward and to make a difference for patients. Effective leadership, again, is a product of a clear vision of what you would like to achieve, being a good mentor and empower your mentees. And I think that at the end of the day, this is all what effective leadership is about. No micromanagement, and a clear vision, and empowering people, and helping them and to support them to grow into positions, even very early in the career. And I've seen fantastic scientists at the pre-graduation level, seeing them thrive and grow is just pure joy. On the matter of institutions, of course, these are important. Institutions that support a certain field, that are maybe strong in a certain field, for instance, neuro-oncology. But I never had the plan to become EANO president, right? So this was always driven by curiosity and interest. Then there comes a stage of your career where you have the desire to bring something to the table, as I have a desire to bring something to the table of this organization within the 2 years of my presidency. And then someone else would take over with new ideas. And this is the way how organizations then thrive. But it's important to always put content before positions. Otherwise, I think effective leadership will not work. If you thrive to become a president of some organization or a leading figure in ABZ, it's less likely to work. If you follow your ideas and a vision, then these things come eventually at some point of your career.
Thank you so much.
Dr. Platten, over the years, neuro-oncology has seen some major shifts from purely surgical and radiotherapy approaches to integrating molecular biology and immunotherapy. How have you seen the field change, and where do you think it's headed in the next decade?
This is a very interesting question. I think that the change in neuro-oncology is a major change from what I've experienced. I've shown you some of the gels that I have done by individually PCIing out a certain region of a gene and doing this sequentially over and over again. I would not be able to give this task nowadays to any medical students, A, because the equipment is not there, and B, they would look at me funny and saying, why don't I just do single cell sequencing and get the information from tens of thousands of cells at the same time? So this is obviously something that has changed dramatically. At the same time, with respect to innovation, neuro-oncology and molecular neuropathology is a good example, has been a driver of innovation. So it's really advancing the field for all oncology. There certainly has been a shift towards additional modalities such as immunotherapy. And I think we still have not, for neuro-oncology, left the dark ages of immunotherapy. But eventually we will overcome this, similar to how it's been overcome for diseases like melanoma. So we will make progress in the future. I think that at the end of the day, this will not be an either or. So either it's immunotherapy or targeted therapy or surgical approaches or radiation approaches. I've seen marvelous advancements in surgical approaches, also radiation approaches. And what we're trying to do is learn from each other and trying to advance the field together. For instance, clinical trials that we do nowadays are, if possible, what we call window of opportunity trials. So these are patients whom we treat with an experimental treatment that are scheduled to undergo a resection. And we postpone the resection in this clinical trial at the cost of an experimental treatment. This necessitates a close interactions with the neurosurgeon. So we wouldn't be able to do this without fantastic neurosurgeons. But then allows us to analyze in detail the tissue of patients who had already undergone previously an experimental treatment. So we can assess the effect of this experimental treatment in the tumor tissue. So this is only possible with close interactions with the neurosurgeons. If you think about ways to administer certain types of therapies, for instance, in the intraventricular space, this is only possible with neurosurgical approaches, wonderful approaches to have an intraoperative profile of tumors, be it with Raman spectroscopy or molecular pathology. So what I'm trying to say is, in a nutshell, there has been advances in all fields across the board, and there's a lot of cross-fertilization between the different fields. And this is one of the reasons I like working with the European Association for Neuro-Oncology so much, because it's really an interdisciplinary, interprofessional organization where neurosurgeons, basic scientists, neuro-oncologists, medical oncologists, and radiation oncologists, neuropathologists come together and exchange ideas and develop new concepts.
Awesome, thank you.
And Dr. Platten, I think I was very curious. You mentioned how LLMs are they're here to make quite a few changes, right, in how we do research and how people just like attain knowledge in general. I guess my question is when you look at your students and peers and your own work, how do you perceive the impact that AI has already had on study and work culture? What are the red flags and what are the good things that come from that?
I think that we're currently at a stage where this can be an enormous help in searching for information, getting structured information and an overview of a certain field. Also, trying to assign certain daily tasks to such large language models. So I think that it's very important to be able to use them, right? Maybe use them in clinical practice. Whenever you have a large array of data, and trying to come up with a meaningful way to prioritize, for instance, differential diagnoses in a very difficult case, or have an array of data where you would like to bring structure in. What the flaws currently are is that you have to check in every detail the output of what you're receiving from such large language models. For instance, if you write a review article on XYZ, these models are very good, at least how I perceive them, in giving you an overview and a structure. but the details are not very good. So these need to be checked. But it's a very good starting point, not to miss out on certain aspects. But then you have to fill in your details. What I think that where the individual scientist will always, not always, but for the next years be more successful is coming up with ideas, the creativity, and also asking the right question. I think that for me as a scientist, this is very important, that I try to ask questions again and again until the answer is I don't know. And then you're at the point where it becomes interesting. When the question really is I don't know. And there are always answers that are I don't know if we dig deep into immunology. And then it becomes interesting. So this is the area where, at least in the current makeup, the large language models are not sufficient. And right, you need to fill this in with your creative mind.
Thank you, sir.
Thank you so much for the insights. Also, going back to your presentation, one of your 10 learning lessons was to empower your mentees. I love to hear how you personally approach mentoring. What are the principles, values, or strategies that you try to pass on to your mentees? And how do you try to create the kind of environment that allows them to thrive both professionally and personally?
So the feedback that I get as a mentor, the most frequent feedback is thank you for giving me the freedom to conduct or follow idea X, Y, Z. And I think that this is for me an important learning lesson is as a mentor, your task is to provide an environment where the mentee, a person, can thrive and follow his or her idea and passion. And this is sometimes a very stringent iterative process. Sometimes it is not. Sometimes it's just quote unquote providing the environment, providing the resources, providing a lab or a department, and saying, I trust you, and then come back when you have questions. What one needs to be mindful as a mentor to see and recognize when someone is struggling. And then you need to have a bit more closer interactions. But at the end of the day, it's the most important, at least for me, task is to instigate this self-esteem, saying, you can do it, you can thrive in such an environment. What matters is follow your ideas. You can discuss and adjust the ideas and saying this is realistic, this is probably less realistic. But then that's about it. And at the end of the day, it is always the task of the mentee to make the most out of this situation. And sometimes during the way, I get the feedback, this is not my kind of thing. I would like to work more in a, I don't know, much more structured clinical environment, or I would like to work much more in a purely scientific environment, and that's perfectly okay. So this is what is, for me, the most important task is providing the environment and the freedom to thrive, and also an empowerment and giving visibility, right? Early on, giving presentations, writing research proposals or papers, because at the end of the day, the career level doesn't matter so much. It's about each and every individual person. And even as a med student or pre-med student, And this was a learning lesson that I had when I was a medical student in Boston and was working with Fred Hochberg, who was my clinical mentor at that time. He made me present at neuro-oncology rounds every week or every other week a certain topic to the neuro-oncologists, radiation oncologists, neurosurgeons as a medical student, and said, next time, give a short presentation on brain metastases and their incidence of bleeding in brain metastases. And coming from Germany and having the task of presenting to authorities and chairpersons of those departments was a daunting task, right? But I managed. And for me, the learning lesson was I sit down, read the literature. Within a very short period of time, you can become an expert on a specific field. And I think that this is very important. If you can transmit this self-esteem and saying you can really do this, you can become an expert in what you're really interested in, irrespective of whether you're a medical student or a postdoc. That for me was an important learning lesson, and this is what I try to also convey and transmit to my mentees.
Thank you so much. So that was our last question for today. Dr. Platten, thank you so much for sharing your remarkable journey and for all the insights you are giving us today. Your thoughts on leadership, mentorship, and the realities of translational research have been incredible, inspiring. We feel truly privileged to have had the opportunity to learn directly from someone who has made such a profound impact on the field of neuro-oncology.
My pleasure. Thanks so much for having me.
Thank you for joining us today. And also on behalf of Med School Mentor, thank you to everyone who joined us today. We wish you all a wonderful day, and we look forward to see you at our next Leaders in Medicine session. Thank you once again, Dr. Platten. Thank you so much.
Thank you. All the best.